Good morning everyone. In this video, we will learn about the replication and segregation of the chromosome in case of bacteria. It is seen in case of E. coli that after the cell elongation, as I have mentioned in my previous video and shorts, there is a replication process begins to double the chromosome. Okay. If this is a chromosome, there is another chromosome will be formed from this one, and the bacterial chromosome will always is circular in nature, which has an origin of replication from where the replication starts, and the replication will terminate it just at the opposite end of the ODC. Okay. So this is the basis of the replication of any kind of a bacteria, specifically E. coli. Then one important thing is, as I have mentioned in case of binary fission, the new chromosome which has been made from the old one, it has to be segregated from its original chromosome, and then it will move towards the end of the cell which has been elongated previously, like this. If it's a bacterial cell, if it's a chromosome, then the cell will elongate, chromosome will be doubled. It gets doubled means replication process starts, and after that what happens, the major process, that is, there is a protein called MreB, or also it is known as murin cluster B protein. What is the function of this MreB protein? The function of this protein is, if it's an elongated bacterial cell, suppose E. coli, Escherichia coli, the MreB protein, it basically acts like a actin protein, and it will form spiral helix, the surrounding of the inside the cell, and the chromosome now has been produced here. Okay. After replication, it looks like this. Now in this elongated cell, what will happen in the next round? This MreB protein helix will pull these two chromosomes at the two ends of the cell, or the two halves of the cell, so that it can give rise to daughter cells or the progeny cells. So it is thought that following this MreB helical formation, the chromosome will move by following this skeletal protein, which actually acts as a actin protein in bacteria, and that will help to move and segregate these two chromosomes at the two ends of the cell, which will give rise to progenies. Okay. So this is all about the replication and segregation of the chromosome in case of E. coli. And there are other bacteria which may follow this kind of a binary fission activities. Hope you like this video. If it's useful, please subscribe my channel. Thank you so much.